Next speaker is Sjoerd van der Burg. Sjoerd is a professor in experimental cancer immunology and therapy at the Leiden University in the Netherlands.
Thank you very much for inviting me. I feel so depressed now. Yes, that's mine. I mean, how can we conquer all these reasons? So I tend to look at it from the positive side and study the immune responses in patients and look at the patients that do well versus the ones that do not so well. Yeah. Thank you. So, if we would look at the tumor microenvironment or the environment of high-grade lesions, what is important, what we know from all these animal models, as well as studies in subjects, patients that do well versus the one that do not, we see that if you have a tumor, and this is the microenvironment, so the tumor stroma within the tumor cells, you will see that these tumor cells start to produce some chemokines like CCL2 and CXCL9, 10, and 11, which attract cells of the immune system. If it all goes well, and this is a situation in which tumors start to regress, or are contained for a very long time, so the people survive for a very long time, you will see that they contain CD4 helper cells that make interferon gamma, or CD8 T cells that have granzymes that can kill off T cells. There are these typical macrophages, and macrophages are known to infiltrate tumors. Usually they try to promote tumor growth, but if you have the type of M1 macrophages, which makes IL-12, and are CD14 positive and CD68, they can also kill via indirect systems tumor cells. The T helper cells can produce interferon gamma, which also upregulates HLA molecules on tumor cells, which are down in many patients. And so this would be a situation which is shown in many different tumors as well as tumor models. This is a kind of protective situation. Either the tumors or lesions regress or the patients survive longer. And also in cervical cancer, we have found that if there's more of these infiltrating CD8 T cells, which can kill tumor cells, then you will have less lymph node metastases. So this is a kind of an improve of survival. So CD8 cells are there. Interferon gamma is also very important, as I said before, because they stimulate these macrophages and they can upregulate HLA class 1 and also HLA class 2 on the tumors. And we recently did a study in which we looked at the interferon gamma production via a Tibet production, the expression of Tibet. And here's an example of a tumor in which all these brown cells, these are all infiltrating lymphocytes, they are all Tibet positives, they all make interferon gamma. On the other hand, we also have tumors which are infiltrated. You can see this dense blue nuclei, which are all lymphocytes, and they don't produce interferon gamma at all. So some of these tumors are infiltrated by lymphocytes that do produce interferon gamma, whereas other patients do not. Now, if you put the disease-free survival curve for those patients, you see that the ones that have a high expression of this Tibet, they really do well in time, whereas the other ones have a quick recurrence. Also, when we look at the macrophages, and this is a recent study, Peggy de Vos van Steenwijk, she will present this on a poster during this conference. If you would look at cells, these cells, and you would quantify them in tumors, and that's what we did in a large group of patients with cervical cancer, you will find these M1 macrophages, and if you find high densities of these cells infiltrating the epithelia of the tumors, you will see that they do much better than the ones that do not. And in fact, this is an independent prognostic factor for survival. So indeed, in HPV-positive tumors, you will find these CD8 cells and these macrophages. You will find this interferon gamma. And also you will find HPV-specific T cells in the blood of such patients. And if they have this expression, if they do have T cells in the blood, this is associated with a longer disease-free survival. So we have this connection of CD4 cells, CD8 T cells, interferon gamma, and macrophages. And this is really doing something good to the tumor. It also means, of course, that this is something we would like to recapitulate when you try to have an immunotherapeutic approach with vaccines. Now, there are many vaccines tested and still being tested. Margaret already talked about the DNA vaccines a little bit. Andreas Kaufmann has worked on the dendritic cells with proteins. There are people working on also DNA vaccines. This is in Connie Trimble's lab and TC Wu. The CanTab vaccines, TG401 and THPV, I don't know whether they still exist because CanTab doesn't exist anymore. You have these proteins which are used by Ian Fraser, E67, Iskometrix, and are peptide vaccines. And I work in a lab in which we test the HPV-16 synthetic long peptides. And this is where I'm going to talk about more because we did many studies with that. And I'm not saying that this is one of the vaccines and the other ones are not good vaccines, which is the one that we studied. And the concept is quite simple. You have an antigen which contains T-cell epitopes in red, green, yellow, etc. And you just cut this protein in large parts of peptides. You synthesize them, you make them overlapping, and you inject them into the skin or subcutaneously. And then these peptides, you can see it like a kind of an apple. If you just take little bites of an apple, you make little parts, you can easily ingest them. And we found that indeed dendritic cells easily ingest these long peptides compared to protein. And if you if they're taken up, they will present these peptide epitopes to CD4 and CD8 T cells, and this was proven to happen, not only in mice systems, but also in humans. And so a couple of years ago already, we tested this vaccine in patients with a high-grade VIN, and it was published 3 years ago in the New England. And what we did is we vaccinated these women 4 times with the vaccine, a high dose of peptide, in montanide, and the outcome was that indeed if you had patients pre-vaccination, these were the lesions on the vulva, after the vaccination you could have partial responses, so there's a little lesion left here, or you could have complete responses. So the response rate at that time was 47% complete response. We already doing a new trial, and I know some of the results. The response rate will be a little bit later, but the point is that indeed here we have a vaccine that can induce a T-cell response, a CD4 and CD8 T-cell response, which has an efficacy to establish lesions. So you can vaccinate, you can get the right T-cell response, and you can get complete responses, but not not in many patients. And so this is the immunological data. I just took 2 of the figures we had in the papers. This is one showing the pre-vaccination response against the vaccine of non-responders, partial responders, and complete responders. And you can see there's a nice incremental response rate to the vaccine. And the best responses are being made by the ones having a complete response. In contrast, all the other patients that did not have a very good complete response, we found that, as said by Margaret already, that these ones did develop regulatory T cells specific for the HPV vaccine. And this is something you have to think about. T cells, they're recognized by the T cell receptor, a antigen, and depending on the milieu these T cells are activated in, they can become effector cells, but they may also become regulatory T cells, or they can be converted from an effector cell to a regulatory T cell. So in this case, these patients over here, they made a response against the vaccine, but with the wrong type of T cell, which can suppress autoimmune cells. You can see the complete response did not have such a response. In fact, they had a very high response of effector cells. So this is indeed a problem. After vaccination, you can get also vaccine-specific regulatory T cells, which can suppress immunity. So if we talk about progress, I looked at the literature last year, and basically there were many articles in the mouse system, and I'm not going to address those. There were just 2 articles in patients. And one is from our own group, in which we tried to vaccinate patients with high-grade CIN3. And the setup was that the colposcopy, they were included, and then they were twice vaccinated, and after 7 weeks, so this is within the normal scheme, they were treated by LEAP. So we had only a very small period to look at what was happening. And the idea was to look for immunogenicity as well as for the infiltration of T cells in the lesion. And unfortunately, we screened a lot of patients, and they didn't want to participate in this trial, mainly because they just want to get rid of the lesion, they want to be treated, and that's it. They didn't bother about science in that sense. So we had quite a problem in recruiting that. But we had 10 patients, and 9 of them were vaccinated. These are the placebo patients, and you don't have to look at the numbers, just look at the gray fillings. You will see that in the placebo group there's hardly any response, but in the vaccine group after 2 vaccinations and in the follow-up after LEAP, you will see that they all made a response to the vaccine. So at least we showed in this trial that the vaccine mediated the immune response, and it was not something induced by either the the treatment of the patients or previous biopsies. And of course, we looked at the infiltration, and in 3 out of 5 we found a dense infiltrate after vaccination of the patients that were vaccinated, and only 1 out of 3 placebos. But this group is so small that you cannot really say anything about that. There was also another trial published from the Cuba group, in which they used just one peptide, one HLA-A2 binding peptide. So this is a peptide that binds in the groove of an MHC class I molecule, and it should be presented on the outside of tumor cells. And they decided to vaccinate with this peptide, as well as with a very small size proteoliposome as an adjuvant. And they looked at clinical responses and immunogenicity in patients with CIN3. They studied only 7 patients, and remarkably 4 of them got a complete response after follow-up. And these are immune data. You don't have to look at the whole table. But the interesting thing was that most of these patients already had an immune response detected by them before vaccination. And this is quite in contrast with what other people see. Mostly we see that these patients do not have an immune response or a wrongly polarized immune response. The other thing that also was noticed from this study is they studied different epitopes we know of, and this is another epitope, was not in the vaccine, and actually the response to this epitope was also increased after vaccination. And so I'm not sure whether this is really a vaccine, a peptide or an adjuvant-related clinical response. I guess that these are patients that either have already a good immune response and are predestined to get a complete response. So after this first result with the VRN, we started also in parallel a study in which we tried to vaccinate patients with a advanced gynecological cancer. These were mostly cervical cancers, but sometimes also an anogenital cancer. And we did the same type of study. Patients were vaccinated just 4 times with 300 micrograms of peptide in montanide. Montanide is just an oil depot from which the peptide leaks for a long time and stimulates the immune response. So this is the immune response of this patient group. You see pre-vaccination, after 2 vaccinations, after 4 vaccination, by different immune assays, measuring proliferation or interferon gamma T cells, the numbers of them, interferon gamma production or IL-5 production. We used interferon gamma and IL-5 because our previous studies showed that these were the cytokines produced in healthy individuals. And what you can see from this is that at least in end-stage patients, so these are the patients which usually have a very poor immune system, and that they can make in response to the vaccine. It's significantly higher than pre-vaccination, but what I know from our other trials is that this response is quite low. The median survival time of this patient group was 12.7 months, and so we decided to divide the patients according to this median survival. And then look at the immune responses. And what you can see here on the left side of this figure is the group with a low survival. On the right side is a group with a relatively longer survival. And what you can see is that the patients that do have a response, they have a high response when they also live longer after vaccination, or when they live longer anyway. We also looked at other antigens, and what we think we see here is that patients that have a better immune response at all, also against other antigens, are also the ones that make a better response. So it's not clear, it's certainly not a proof that the vaccination indeed led to a longer survival time. And to show that even better, so we constructed a cohort, a matched cohort, the same number of patients with the same type of disease, same stages, etc., etc. The whole group was not significantly different from our vaccinated group. And we plot the survival curves, and you can see they completely overlap. So this again indicates that although we see a better response in patients that live longer, the overall survival is not different from a non-vaccinated group. So in this sense, this is quite a negative result, in the sense that we do get an immune response, but we do not get any clinical efficacy, although we use a vaccine that has shown already to be clinically effective in the high-grade lesions. So I come back to this slide again, pointing out what is really good in the immune response against tumors and what should be there. But if you really study tumors and you look at all the data, you will actually find this type of immune response in those tumors. And so it's very complicated. It's a very complicated slide also. But what it means here, you have the myeloid-derived suppressor cells, which is a kind of immature myeloid population that is also suppressing immunity. We have type 2 macrophages there. The type 2 macrophages induce vascularization. They attract also Tregs. They produce IL-10, so they are suppressive. And they can actually suppress the immune responses, as you can see by all the red lines. We also find T cells expressing the inhibitory receptors. These inhibitory receptors usually suppress the function of these T cells. So that's not really helping out. We find regulatory T cells, we find tolerogenic DC, dendritic cells that stimulate the wrong type of T cells. And all this together determines whether you are a good responder or a bad responder. It's really the amount of cells you find. So the numbers between this and this type of macrophages, the type of T cells, effector T cells versus regulatory T cells, that balance within the tumor microenvironment actually results in a longer or a less long survival. And so when you talk about vaccination, this is the only part we are addressing in this particular part. So if you have a vaccine, you can induce CD4 and CD8 T cells. You can also try to give adoptive T cell transfer. So you culture the T cells in the lab before you transfer them back to the patient. So it's all to enhance T cells. And this is also what vaccines only do. They enhance the number of T cells that you really need to target the tumors. But of course, there are many other things wrong in this tumor microenvironment. So you also have to address the expression of these inhibitory receptors. And for that, you can maybe use antibodies. Our antibodies against CTLA-4. It's called Papillomavirus. It's used now in melanoma. It's a treatment. We should also try to use this in cervical cancer. Also, new reports in New England about the antibodies against PD-1 program. That cell 1 is also an inhibitory receptor. This one should also be targeted within the tumor microenvironment, because we know that the T cells, about 50% of the T cells that infiltrate tumors are expressing this type of molecule. Furthermore, we have to try to address the regulatory T cells. There are some antibodies, for instance, in transplants, they use antibodies against CD25, which is a marker also on regulatory T cells. Antibodies like daclizumab, you can try to use those to deplete the CD4 regulatory T cells. Or you may try chemotherapy, not chemotherapy that you would use to treat a tumor, but for instance cyclophosphamide, which is known to suppress regulatory T cells. So you should add another chemotherapeuticum which is not directly targeting the tumor cell, but is targeting a specific part of your immune system. Furthermore, we have to take rid of these M2 macrophages. You can try to use this by thymidine kinase inhibitors. Tumors express COX, prostaglandin E2, the IL-6, and these are known to drive these tumor-promoting macrophages. And by blocking them, for instance, by COX inhibitors or by antibodies against IL-6 receptor, tocilizumab, which is used in rheumatology to treat rheumatoid arthritis, you can try to kill off this particular part of the macrophages. And of course, in the end, you have also to target the tumor by chemotherapy, just to kill off tumor cells, but maybe also particular types of chemotherapy that stimulate the immune system, and they are heavily studied these days. But you may also try to give interferon alpha or interferon gamma just to upregulate processing of these and upregulation of HLA class 1 and 2 molecules and the processing of these viral peptides in these molecules so that they can be recognized by T cells. And basically, if you want to do immunotherapy of cancer, you probably have to take all these parts all together. So this is really something for the clinicians to try to figure out the type of protocol setting in which you can try to not only vaccinate, but also use your chemotherapy, use your antibodies, do it all together and try to get rid of these inhibitory systems. Now we are trying a little bit already in our hospital. This is based on some mouse work, and it's the only mouse work I will show. And this is the typical tumor used for HPV vaccines. These are mice that are non-treated. These are mice treated by cisplatin or carboplatin, so these are chemotherapeutics used in the clinic. These are mice that have been vaccinated. You can see they all die in the end. And these are mice that have combined chemotherapy and vaccination, and suddenly these start to survive. So chemotherapy and vaccination can actually act together. They synergize. Usually chemotherapy is thought of to kill off the immune system. This is not the case. Some of these chemotherapeutica will do this. Others will have different effects. And we are really studying this now in more detail. And not only we, but also others. And you will see that many different chemotherapeutics will have many different effects. Some of them targets macrophages, the other one targets regulatory T cells, and some just kill off all immune cells. But this is really a field of study that is now growing. And you can see, indeed, if we vaccinate, we have more T cells in these mice. And also, when you combine them with chemotherapy, you will see the same number of T cells. So there's not more T cells, just the same number. So there's no negative effect. There's also not a bad effect. So this chemotherapy has an other effect, not affecting T cells. And we took this to the clinic. And this is quite a complicated slide, but you don't have to look at the details. So what we did is we had end-stage patients which were treated with carboplatin and chemo and paclitaxel. And this is how the macrophages subtypes look in a healthy subject. So just focus on this, all the different in red and blue and in black. And this is a patient before it got chemo. And what you can see is that this population, the red population, is increased from 7% to 22%, which is a real high increase in myeloid cells, which are suppressive. You also see that there's a decrease in this blue population, and not given in percentages, but you can also see it over here. So here's a void, which is here, again, black, and these are dendritic cells, actually. Now, after one chemo, there was nothing happening, but after 2 chemos, we saw that indeed this population of cells suddenly normalized, similarly to what we see in healthy subjects. So this chemotherapeutic did something with our macrophages, and they just brought it back to the normal situation. And this was observed in a couple of patients. And so we decided also to look at the immune responses, and we found out, and it can be seen here in the white and black bars, that after the second chemo there was a better response of the capacity of the antigen-presenting cells to drive T cell responses, and also T cells against viral and bacterial antigens had a much better response rate. So there was a positive effect on the T cells by just getting away or normalizing the myeloid cell population by chemotherapy. And based on this, we decided to move on and to vaccinate patients during their after the second chemotherapeutic line. And this is the result. So these are just 4 patients. We are still doing this. We're now on 10 patients already. And what we see is basically that pre-vaccination, there's no response. After vaccination, there's a huge response. This is the whole vaccine. You can see that almost the whole vaccine is really recognized. And in particular, this patient, we already have data after all 6 cycles of chemotherapeutics. And you will see there's no difference in the response. And what we see is that indeed we get a much bigger immune response just by giving the patients chemotherapy, and this response is not affected by continuous cycles of this chemotherapy. So this is where we are now, and we try to start next year a large trial in which we will vaccinate patients during the chemotherapy, and we will see whether we get some effective data. So these are all the people involved, and I stop here. Thank you.